In this mini lecture, we're going to go over the adaptive immune response, sometimes called the acquired immune response. This encompasses our body's ability to recognize and defend itself against invaders. There are 5 attributes that you want to remember that characterize adaptive immunity. The first is specificity, meaning our body learns to respond to a particular invader on an individual basis. This means that once you respond to something, your immune system will recognize that exact or nearly exact pathogen and effectively respond the next time. If the pathogen is not closely related, the immune response will not respond as quickly, but will start to learn how to respond to the new pathogen. It is inducible, meaning your immune system learns how to respond on a first encounter, and in subsequent encounters, you turn on a quick response. The clonality is referring to the fact that we're going to be stimulating specific white blood cells, in particular our lymphocytes. The stimulated lymphocytes are going to make copies of themselves. In other words, they will clone themselves to expand the numbers that are capable of effectively responding to a specific pathogen. We'll talk more about that as we go along. Also, a functioning immune response should not respond to self, only to foreign molecules, so it's unresponsive to self. And finally, it has a memory. This is the benefit of learning to respond specifically. Once learned, the memory allows a quick response the next time. Your immune response will remember the invaders seen previously. Now, as you look at the graphic at the bottom, we'll talk about these different scenarios. If we look overall at immunity, we have talked about our innate or non-specific immune response already. Now we're covering the adaptive immune response, and we're going to be looking at both the natural and the artificial types of stimulation of the immune response. The natural immune response, as you might imagine, is a response that happens naturally. You can naturally get acquired immunity by experiencing an active infection. Or an infant can experience passive immunity when mom passes her antibodies to a fetus through the placenta, which remains in the bloodstream for some time after birth, or through transfer of antibodies in breast milk. Artificial immunity is encompassing things like immunization. This stimulates an active response from the individual, and memory will develop. Also, when we give somebody antibodies as treatment to protect them temporarily against an invader, this is passive and temporary. No memory will develop here because no cells are involved. Antibodies are proteins and they degrade over time. We're going to be spending most of our time talking about lymphocytes. When we talk about lymphocytes, and one is shown in the figure up in the corner, we have B lymphocytes or B cells, and we have T lymphocytes or T cells. B cells, it's kind of easy to remember where they mature because all blood cells are formed in the bone marrow, and B cells actually mature there. B for bone. T lymphocytes start in the bone marrow, like all the other blood cells do, but they go to the thymus to mature into T cells. So they grow up and are schooled in the thymus. T cells mature in the thymus. We also have a type of lymphocyte called the natural killer cell. These were mentioned as part of our innate immune system. Here, in this section, we'll talk a little bit about them and their role in adaptive immunity. Our adaptive or acquired immune response has 2 branches. So we have humoral and cell-mediated adaptive immune responses. And in this section, we'll be talking about these 2 branches and the major players within them. When we look at the tissues and organs of the adaptive immune response, the lymph system is extremely important. You can look at this figure here on the left, and you can see the color code of different organs and tissues that are involved in the lymphoid system, including our lymph nodes, Peyer's patches, adenoids, and appendix. These are places within the body that white blood cells tend to congregate. You might not have heard of MALT or GALT before, but they are important for protection against invading microorganisms. MALT stands for mucosal associated lymphoid tissue, MALT, and GALT is gastrointestinal associated lymphoid tissue, GALT, which is really a subset of MALT. These are areas of congregating white blood cells, not as organized as something like a lymph node, which we'll be looking at in a minute, but they are more like loose congregations of cells that hang out in an area to try to protect us. Do you remember that we spoke how many pathogens find it easier to invade the mucosal tissues compared to skin? Well, these are the white blood cell sentries that are attempting to provide a level of protection against those invaders in this more susceptible tissue. Let's look at a lymph node now. This figure from your book is showing the anatomy of a lymph node, and you should find it helpful to understand it. You'll see that we have lymph vessels that are feeding the lymph node. These vessels have collected the extracellular fluid from the tissues. So these vessels are draining the tissues and contain what is now known as lymph fluid. The lymph is flowing through the lymph node. It must percolate through and come out over here at the efferent vessel. So all that lymph fluid from the tissues that may have picked up any antigens, bacterial, viral, whatever, is going to trickle through that lymph node. And remember, we have all kinds of white blood cells hanging out in the lymph node, and this is where we can get activation of our immune response. All the purple areas that you see here are representing what they call germinal centers, where we have activated lymphocytes and immune cells working together, getting ramped up and ready to fight off invaders that have been detected while they moved through our tissues and lymph system. Now, because this gland is all tissue, it needs to be fed. So we have a blood supply that is feeding the lymph node through these vessels. We have an arteriole bringing in blood and venules that are draining out. This draining venule will merge with larger and larger veins, and the draining lymph vessel will eventually rejoin the circulatory system. So make sure you have a look at the lymph node and understand what's going on here as antigens percolate through. And as we talk more about activating our immune response, remember that there are different cells in here interacting with each other in close quarters, which makes it much easier for cells that need to talk to each other to actually do so. Now let's talk about antigens. Earlier, I used that term, and perhaps you've heard that terminology before. We define antigens simply as something that our immune system can respond to. Antigens are often proteins or parts of proteins, but they do not have to be. They can be other types of molecules like carbohydrates or other. At the same time, I want to draw your attention to this term right here, epitopes. Epitopes are the specific regions of a larger antigen that is going to be recognized. So if you have a large protein, only a little part of that would be recognized by any particular cell type that can see and respond to it as an antigen. That portion of an antigen that is actually the recognized portion is the epitope. Let's now talk about the types or classes of antigens. We have antigens that exist outside of our own cells. Those would be exogenous antigens, right? Exo for out, outside of our cells, or extracellular. This would include things like a virus particle floating through our tissues, or a bacterial cell that got introduced into a surgical site or a wound. The other type of important antigen is endogenous. Endogenous antigens are intracellular or inside the cell. Imagine a cell that may be infected with the virus and the virus is inside, or it could be a bacterium inside the cell. These are endogenous. We need to keep those two things separate because they are important to activating our immune response. We are not going to be talking about it in this chapter, but in a future talk, when we discuss how the immune system might go awry and not function correctly, then we will discuss autoantigens. Autoantigens are normal or self-antigens that our immune system is inappropriately responding to. But that's not really the topic of this chapter. Exogenous and endogenous antigens are. Another key player in this whole complex dance of our immune response is what we call the major histocompatibility complex, or MHC. This set of molecules helps our immune system respond. These are glycoproteins that are found on the surface of cells, and one type is found on most cells. The other type is found on a more narrow group of specific cells. So we have these two types or two classes of MHC. Class 1, or MHC class 1, is on most nucleated cells of our body. It's responsible for interacting with our cytotoxic T cells. Specifically, the interaction is between the cell with the MHC1 and the right epitope and the T cell receptor of the cytotoxic T cells. We'll talk about that shortly. The MHC class 2 is only on specific cells. Only a subset of cells in our body have it, and they include B cells and phagocytic cells like dendritic cells, macrophages, and others. Keep those two ideas in your mind as we're moving through this section. Here's a picture from your book, so a reminder that every nucleated cell in our body have class 1 MHC, but that would exclude red blood cells, RBCs, because they lack a nucleus. We have a picture here with membrane receptors that have a spot to grab an epitope. Class 2 MHC, this one's shown in lighter blue, also has an antigen binding site. You can see it is shaped just slightly differently, but very similar to MHC1. And a cell that has MHC2 would also have MHC1. So this cell has both MHC2, the light blue one, and the MHC1, the darker blue one. Keep that in mind. Well, how do you get MHC to grab an epitope? Well, there are two pathways. You might imagine that because you might recall we have two different types of antigens and two different classes of MHC. If it's an exogenous antigen, and remember, those came from outside, a cell would have to phagocytose it. You see this little red ball representing some sort of cell type that is getting ingested or phagocytosed. Then the phagosome and lysosome fuse, allowing MHC2 to grab an epitope. And as a result, when the cell does exocytosis, then MHC2 and that epitope will be displayed out on the cell surface with that piece of the phagocytosed antigen being presented on the cell surface. The immune response will be stimulated, specifically our helper T cells. This is our exogenous processing pathway. For the endogenous processing pathway, it deals with epitopes that are found on the inside of the cell, an internal epitope. For example, something like viral messenger RNA instructing the cell to make viral proteins. The viral protein in the internal area of the cell will then be sampled. All cells do a continuous check of what is going on inside and have a normal sampling mechanism that occurs. This lets the surveillance team, the cytotoxic T cells, see what's going on. Internal molecules are continually put out on the cell surface with MHC1. So now we have MHC1 with this internally produced protein, which in this case is the viral protein. Cytotoxic T cells see this and will be activated to eliminate the cell showing foreign epitopes with MHC1. This last figure has a little more detail. You can see that we have a virus that came inside the cell, and the cell made viral proteins. The cell has a sampling mechanism that is going to be sampling some of the internal proteins. That sampling mechanism joins epitopes with MHC class 1 and puts it back out on the surface. There, it is going to be interacting with our killer T cells, the cytotoxic T cells. For the exogenous antigens, they are endocytosed. We have a phagosome and a lysosome that are going to fuse. That is going to allow the digested up pieces of the exogenous antigen to be coupled with MHC2 and ultimately to be put back out onto the surface. There it will interact with helper T cells. So while this figure has more details, it's showing the same thing as that more simplified figure shown earlier. That is going to be the end of this first lecture on adaptive immunity.